Welcome back, everyone, for another deep dive. This time, we're taking a closer look at RNA. It's so much more than just a messenger between DNA and proteins.
It really
We've
is.
got some pretty dense sources today.
Definitely dense.
But I think that's great for our listeners because they're ready to take all this in. I know I'm excited to learn about RNA acting as an enzyme,
Oh, yeah.
and even how RNA could have been involved in the origin of life itself.
Absolutely. RNA research is such a dynamic field. There are constantly evolving discoveries that really challenge how we think about life.
So to start, can you walk us through the synthesis of ribosomal RNA? It's got to be a crucial part of building the protein synthesis machinery in a cell.
Right. So the synthesis of that rRNA starts with RNA polymerase and
Oh.
transcribing those RNA genes. In eukaryotes, this is all happening in the nucleolus, so that's a specific area inside the nucleus. RNA polymerase high is specifically going after that 45S pre-RNA gene, and it makes the 45S pre-RNA transcript.
Okay, so we have this pre-RNA molecule, but it's not ready to go just yet, is it?
Nope, not quite yet.
It needs to go through some pretty fascinating modifications before it can become part of the ribosome.
Exactly. That 45S pre-RNA goes through a series of steps. One of the key ones is methylation. That's where methyl groups are added to very specific nucleotides in the pre-RNA, and those methyl tags are like signals for the next step, cleavage.
So it's kind of like a molecular sculptor comes in and chisels away at the pre-RNA,
Yeah.
taking off the parts that aren't needed to reveal the functional parts.
I like that analogy. It's a great way to think about it. So specific endonucleases cleave that pre-RNA at precise locations. They're using those methylation tags to guide them. And from that, you get the mature 18S, 5.8S, and 28S RNA molecules,
Yeah.
which are all essential components of the eukaryotic ribosome.
But there's that fourth RNA we can't forget about, that 5S RNA.
You got it. And that one is actually transcribed by a different enzyme, RNA polymerase III. Unlike those other rRNAs, the gene for the 5S RNA is located outside the nucleolus, but once it's transcribed, also goes through some processing, although it's not as extensive as the 45S pre-RNA.
So once we have all four of those RNA components synthesized and processed, they're ready to team up with the ribosomal proteins
Right,
and
yeah.
form the ribosome, the protein-making machines.
Right, absolutely.
All right, so now let's compare RNA transcripts in prokaryotes and eukaryotes, two very different cell types.
Yeah, so in prokaryotes, they don't have that membrane-bound nucleus. So transcription and translation happen at the same time in the cytoplasm. Prokaryotic mRNA is often polycistronic, meaning that a single mRNA molecule can code for multiple proteins.
So like that mRNA is multitasking. It's got the blueprints for multiple proteins all in one.
Exactly. Now, in eukaryotes, their mRNA is usually monocistronic, meaning it only codes for a single protein, and it goes through some processing steps before it can be translated.
Like splicing, where those non-coding introns are removed.
Exactly. Splicing is super important in eukaryotic mRNA processing. So you've got the introns, those are the non-coding sequences, and they're removed from that pre-mRNA. And the exons, the coding sequences, they're joined together to form the mature mRNA. And this whole process is done by a complex called the spliceosome.
I also remember learning that eukaryotic mRNA gets a cap and a tail.
Yes. You're talking about the 5-foot cap and the 3-foot poly-A tail. Those are two key modifications that protect the mRNA from degradation and enhance its translation. So the 5-foot cap is a modified guanine nucleotide. It's added to the beginning of the mRNA. And the 3-foot poly-A tail is a string of adenine nucleotides added to the end.
So it's like the eukaryotic mRNA gets the VIP treatment before it heads off to the ribosome for protein synthesis.
Uh,
All right. I think it's time to dive into a concept that really challenges the traditional flow of genetic information.
I agree.
RNA-dependent RNA and DNA synthesis.
Right. This is where we really see how versatile RNA is. So the central dogma of molecular biology says that DNA is transcribed into RNA, and then RNA is translated into proteins, but RNA-dependent synthesis shows us that RNA can also be used as a template to make more RNA, or even DNA.
And the best example of this is with retroviruses.
Yeah, absolutely. Retroviruses like HIV are RNA viruses that use reverse transcriptase to convert their RNA genome into DNA. Then that viral DNA integrates into the host cell's genome, which lets the virus replicate and persist.
Reverse transcription is such a remarkable process. It basically rewrites the rules of molecular biology. It shows that the flow of genetic information isn't always a one-way street. Can you walk us through the steps of reverse transcription?
Yeah, so it starts when the retrovirus enters a host cell and releases its RNA genome. Then the viral enzyme reverse transcriptase uses that RNA as a template and synthesizes a complementary DNA strand, or cDNA.
So now we have a single-stranded cDNA molecule, but it needs to pair up to integrate into the host's DNA.
You got it. And that's where the dual nature of reverse transcriptase comes in. It can actually switch from an RNA-dependent DNA polymerase to a DNA-dependent DNA polymerase. So it uses that newly made cDNA strand as a template to create a second complementary DNA strand. And now you have a double-stranded DNA molecule that's ready to integrate into the host cell's
Huh.
genome.
It's incredible how one enzyme can have two distinct functions, like it's orchestrating a molecular takeover. But I remember learning that reverse transcriptase is a little sloppy, doesn't have that proofreading ability like other DNA polymerases.
That's right. That's a key characteristic of reverse transcriptase, and it's a big deal for retroviruses. That lack of proofreading leads to a high mutation rate, which makes it really hard to develop effective treatments, and it also contributes to drug-resistant strains emerging.
It's like a molecular game of chance. With every replication cycle, there's a chance for a new mutation that could change how the virus behaves.
Exactly, and that constant genetic variation is a huge challenge in fighting retroviruses.
So let's move on to another example of reverse transcriptase, the enzyme telomerase.
All right.
Telomerase protects our chromosomes. I'm really interested to learn more about this enzyme and how it's connected to aging.
Yeah, so telomerase is super important in keeping our chromosomes intact. It adds repetitive DNA sequences to the ends of chromosomes, called telomeres.
And those telomeres are like the protective caps on our chromosomes that keep them from fraying and unraveling.
Exactly. That's a great way to think about it. Every time a cell divides, the telomeres get a little shorter. It's a natural part of aging. But some cells, like germ cells and stem cells,
Hmm.
have high telomerase activity, which stops the telomeres from shortening and lets those cells divide indefinitely.
It's like telomerase is a fountain of youth for those special cells,
Well, yeah,
keeping them young.
you could think of it that way. But there's also a downside to telomerase. In most cancer cells, telomerase is reactivated, and this lets those cells escape the normal limits on cell division, so they proliferate uncontrollably.
That's fascinating. So telomerase plays a part in both healthy aging and cancer.
That's right.
Okay, now I think we're ready to enter the world of ribozymes.
All right, get ready for RNA with a hidden talent.
Huh.
Ribozymes are catalytic RNA molecules, which means they can speed up chemical reactions just like protein enzymes when they were discovered. It completely changed how we thought about RNA and what it can do.
So they're like superheroes, wearing a messenger cape, but secretly wielding enzymatic powers.
I like that. One of the most studied ribozymes is the hammerhead ribozyme. It gets its name from its shape. You find this one in certain plant viruses, and it can cut RNA molecules at specific spots.
Like molecular scissors.
Exactly. Another one is the self-splicing intron. These are found in some RNA and tRNA molecules, and they can actually remove themselves from the pre-RNA molecule without any help from other enzymes.
That's incredible. It's like they have a mind of their own. Ribozymes really challenge how we think about the roles of RNA in the cell.
Sure.
Now, I want to talk about the implications of ribozymes for the origin of life. The RNA world hypothesis. Are
Absolutely.
we ready to dive into that?
Let's do it. The RNA world hypothesis basically says that RNA was the main carrier of genetic information in early life, not DNA. It's a fascinating idea, and it still causes a lot of debate and research.
That's what I love about the RNA world.
Yeah.
It's always full of surprises, and it really pushes what we know about the fundamental processes of life.
I agree.
Okay, so let's travel back in time to a world where RNA might have been in charge.
Sounds good to me. So the RNA world hypothesis proposes that early life forms used RNA for both inheritance and catalytic functions. It's a lot simpler than the system we see today with DNA, RNA, and proteins.
So how did we go from that RNA world to the DNA world we have now? What steps happened in that evolutionary journey?
Okay, so picture this. You've got the primordial soup, and it's filled with all these simple organic molecules like nucleotides.
Building blocks of RNA.
Exactly. And through random chemical reactions, those nucleotides start joining together, forming these short chains of RNA. And some of those RNA molecules, they might have folded into shapes that actually gave them catalytic activity, like the ribozymes we talked about.
Yeah. So now we have these ribozymes that could make even more RNA molecules. It's like a self-replicating system.
Exactly. And that self-replication is a key part of the RNA world hypothesis because it lets genetic information be passed on, even without DNA.
Okay, so we have RNA replicating,
Yeah.
catalyzing reactions, it's running the show. But when does DNA come in?
That's a great question, and it's one that scientists are still trying to figure out. One idea is that DNA came about because it's a more stable way to store genetic information. RNA is less stable, and it breaks down more easily.
Right, that 2-beta-hydroxyl group on the ribose sugar in RNA makes it more reactive.
You got it. But DNA, with its deoxyribose sugar. Doesn't have that hydroxyl group, so it's much more stable. It makes sense that DNA would eventually take over as the main way to store genetic information for the long term.
So now we have DNA as the archive for genetic information, and RNA is taking on those more specialized roles, like messenger and catalytic functions. But where do proteins fit into all of this?
Well, the RNA world hypothesis suggests that those early ribozymes, they might have actually started to make short peptides, the building blocks of proteins.
So RNA is making proteins, and then those proteins can start to do more of the complex catalytic jobs, freeing up RNA to do other things.
Exactly. It's a really cool example of coevolution, RNA and proteins working together to create more complex life forms.
The whole RNA world idea is mind-blowing. It's like we're rewinding the tape of life and seeing a totally different starting point.
Yeah.
But are there any problems with this hypothesis? Are there any scientists who aren't convinced?
Oh, of course. Like any scientific theory, the RNA world hypothesis has people who disagree with it, and there are still things we don't know. One of the biggest challenges is figuring out how those first nucleotides and ribozymes even came about.
It's a chicken and egg situation, right? You need RNA to make more RNA. But how did that very first RNA get there?
Exactly. Another challenge is explaining how those early ribozymes were complex enough to do all those important jobs. It's a puzzle that scientists are still working on.
It's amazing to think about all the possibilities and all the research going on to try and solve the mysteries of the RNA world. It reminds us that we're still exploring and debating even the most basic parts of life.
I agree.
Okay, so let's go back to that RNA synthesis process
Yeah.
and focus on the specifics within eukaryotic cells.
All right. In eukaryotes, RNA synthesis happens in the nucleolus. It's a special area inside the nucleus where the RNA genes are transcribed and the ribosomal subunits are put together.
The nucleolus, I remember learning about it, but I always imagined it as this mysterious thing hiding in the nucleus.
It really is fascinating. You can think of it as the ribosome factory for the cell. It's constantly making those crucial protein synthesis machines.
That makes sense. Ribosomes are so important for a cell to work, so it's no surprise that eukaryotic cells have a dedicated space to make them. Can you walk me through how RNA synthesis works in the nucleolus? I want to get a really clear picture of the whole process.
Okay, so the genes that code for rRNA in eukaryotes, they're all clustered together in specific regions on certain chromosomes, and those regions are called nuclear organizer regions, which is a pretty fitting name.
Yeah, so we have these RNA genes all lined up, ready to be transcribed. Which RNA polymerase is responsible for that?
It's mostly RNA polymerase that does the job. It transcribes most of the rRNA genes and makes a large precursor molecule called the 45S pre-RNA.
45S pre-RNA, that sounds familiar. It's the precursor for 3 of the 4 main RNA components in eukaryotic ribosomes, right?
That's right. The 45S pre-RNA is processed to make the 18S, 5.8S, and 28S RNAs.
So one pre-RNA molecule makes 3 different RNA components. That's really efficient. What about the 5S RNA? Does that come from the same pre-RNA molecule?
No, the 5S RNA is transcribed separately by RNA polymerase III, and the genes for 5S RNA are actually located outside the nucleolus.
Interesting. So there are 2 different RNA polymerases working together to create all 4 RNA components. And then there are those processing steps, like methylation and cutting. Do those also happen in the nucleolus?
Yeah, the nucleolus is where it all happens. After the 45S pre-RNA is transcribed, it goes through all those steps, including methylation and cleavage, to create the 3 mature rRNA molecules.
Can you remind me how those methylation tags work again? They're like the markers for the cutting enzymes, right?
Exactly. Special enzymes called methyltransferases, they add methyl groups to specific nucleotides in the pre-rRNA molecule, and those methyl groups act as markers. They protect those regions from being cut and guide the enzymes to cut in the right places.
So it's like a molecular origami project, with the methylation tags as the fold lines, guiding the cuts to make the final shape.
I like that analogy. Once all the cutting is done, we have the 3 mature RNAs, the 18S, 5.8S, and 28S RNAs.
But what about the 5S RNA? Does it need any processing?
It goes through some processing too, but it's usually not as much as the 45S pre-RNA.
So now we have all 4 RNA components. They're synthesized, processed, and ready to join up and form ribosomes. Where does that assembly happen?
You guessed it. It happens in the nucleolus, the ribosomal proteins, which are made in the cytoplasm. They're transported into the nucleus and then into the nucleolus. And that's where they combine with the mature RNAs to make ribosomal subunits.
So the nucleolus is like this busy construction site. with RNA molecules and ribosomal proteins all coming together to build those protein synthesis machines.
Exactly. And once the ribosomal subunits are assembled, they're sent out of the nucleus into the cytoplasm. That's where they can finally get to work translating mRNA into proteins.
It's amazing how all these different parts work together. It really makes you appreciate how complex and efficient eukaryotic cells are. All right, now let's shift gears a bit and talk about RNA-dependent RNA and DNA synthesis.
Sounds good.
We talked about it a little before, but ready to go deeper. It's fascinating how RNA can break away from that traditional central dogma of molecular biology.
Definitely. So let's start with RNA-dependent RNA synthesis. It's where an RNA molecule is used as a template to make another RNA molecule. And it's all done by an enzyme called RNA-dependent RNA polymerase, or RDRP.
So it's like RNA is making a copy of itself, and RDRP is the copy machine.
Exactly.
Where do we see this happening?
Well, a good example is in RNA virus replication. A lot of viruses, like the ones that cause influenza, measles, and polio, have RNA genomes.
Right, RNA viruses. I remember learning that they're often harder to treat
Uh-huh.
because they mutate so quickly.
That's right, and that rapid mutation is often because of RDRP. Unlike DNA polymerases, which have proofreading mechanisms, RDRP doesn't really have that, so it makes more mistakes during replication.
It's like a game of telephone, where the message changes a little bit each time it's copied.
I like that analogy. And those changes, those mutations, can change the virus's proteins, so it can evade our immune system or become resistant to drugs.
So to develop good antiviral therapies, we need to understand how RNA-dependent RNA synthesis works.
Absolutely. Now let's talk about RNA-dependent DNA synthesis. That's where we have reverse transcription. And the flow of genetic information goes backwards, from RNA back to DNA.
And that's where reverse transcriptase comes in.
Exactly. Reverse transcriptase is the key to making that happen. It can act as both an RNA-dependent DNA polymerase and a DNA-dependent DNA polymerase.
Can you remind me how those two functions work to make a DNA copy from an RNA molecule?
Sure. Let's say a retrovirus, like HIV, infects a host cell. The virus carries its genetic information as RNA. But to get into the host cell's DNA and take over, it has to convert that RNA into DNA.
It's like the virus is trying to sneak its own book into the host cell's library.
That's a good way to put it. So the first step is that reverse transcriptase uses its RNA-dependent DNA polymerase activity. It uses the viral RNA as a template to make a complementary DNA strand called cDNA.
So now there's a single strand of cDNA, but it needs to pair up to integrate into the host's double-stranded DNA.
You got it. And that's when reverse transcriptase switches to its DNA-dependent DNA polymerase mode. It uses that first cDNA strand as a template to make a second complementary DNA strand. And now you have a double-stranded DNA molecule, ready to be integrated into the host cell's genome.
It's amazing that one enzyme can do both of those things. It's orchestrating this whole molecular takeover. But reverse transcriptase doesn't have that proofreading ability.
Right. And that lack of proofreading is a big problem for retroviruses. It causes a much higher mutation rate than regular DNA replication, which makes it hard to develop treatments.
It's a constant battle, trying to stay ahead of the virus as it mutates.
Exactly. This is why it's so important to understand how reverse transcription works, so we can come up with new ways to fight retroviral infections.
This has been such an incredible journey. We've covered so much, from the RNA world hypothesis to ribozymes and reverse transcription. I can't wait to see what else the world of RNA has to offer.
There's still so much to learn about RNA.
It feels like we've only just started.
Yeah.
What else can you tell us about the potential of RNA research?
Well, one really promising area is RNA therapeutics.
RNA therapeutics, what's that?
So scientists are exploring different ways to use RNA molecules to treat diseases. One way is to use something called siRNAs, small interfering RNAs, to basically silence certain genes.
siRNAs. So you're using RNA to target a specific gene and turn it off. What kind of diseases could that treat?
A lot. siRNAs could be used to target genes that are involved in cancer growth, or viral replication, or even genetic disorders that people inherit.
Imagine being able to silence a gene that's making a protein that's not working right, and then the body could make a healthy version instead.
Hmm.
That's amazing, like you're rewriting the genetic code with RNA to make someone healthy.
Exactly. And then there's mRNA vaccines. We've already seen how well they work against COVID-19.
Right. Those vaccines deliver mRNA molecules that code for viral proteins into our cells, and then our cells make those proteins,
Yeah.
which triggers an immune response.
Yeah. It's like giving our immune systems a preview of the virus so they can be ready to fight it off. The success of those mRNA vaccines has opened up so many possibilities for fighting other infectious diseases, too.
Are there any other diseases that mRNA vaccines could work against?
Researchers are looking into that right now. They're exploring mRNA vaccines for things like influenza, HIV, and even malaria. And beyond infectious diseases, they could even be used to target cancer cells by training the immune system to recognize and destroy them.
Wow. The potential of RNA therapeutics seems huge. It's like a whole new world of medicine. And RNA is leading the way.
I agree. It's exciting.
And RNA research isn't just about therapeutics, it's also helping us answer some of the biggest questions in biology, like how life even began, that RNA world hypothesis. I'd love to learn more about the latest research in that area.
Sure, scientists are always looking for more evidence to support or challenge the RNA world hypothesis. One thing they're doing is trying to recreate the conditions of early Earth in the lab to see if they can make RNA molecules from simpler building blocks.
So it's like a time machine, taking us back to the primordial soup to see if RNA could have just appeared.
Exactly. And other researchers are studying ribozymes to see if they could have done those essential jobs in early life. They're even designing and making new ribozymes, pushing the limits of what RNA can do.
It's incredible that we can manipulate RNA so precisely now, like we're molecular architects.
Yeah.
This deep dive has been amazing. We've learned so much about the different roles RNA plays,
Yeah.
from its basic job in protein synthesis to its potential as a medicine, and its possible connection to the beginning of life itself.
It really shows how important RNA is. And the field is constantly changing with new discoveries happening all the time.
For our listeners, I encourage you to keep learning about RNA. You never know what you might discover.
I agree. Keep asking questions, keep learning, and keep exploring the natural world.
Thanks for joining us on this deep dive into the world of RNA. We'll see you next time.